I'm Marta Fernández Matarrubia, clinical neurologist at Marqués de Valdecilla University Hospital, and it's a real pleasure to be here and chair this session. Today, we'll be discussing the evolving role of glial cells in Alzheimer's disease and other neurodegenerative conditions. The goal of this session is to challenge the traditional neurocentric view of the disease and show how glial cells are important not only in neurodegeneration but also in amyloid and tau pathology. The format of this session will consist of 4 25-minute talks by Dr. Estrella Morenas, Dr. Amaia Arranz, and Dr. Eduardo Zimmer and myself, followed by a 20-minute discussion panel, so please hold your questions until the end of the presentations. I will start with the first talk, giving an overview of glial cells in Alzheimer's disease, from basic mechanisms to biomarkers and new targets. These are my disclosures. As you know, the link between neuroinflammation and Alzheimer's disease is not new. In fact, Alzheimer's himself in 1911 described the presence of hypertrophic glial cells surrounding amyloid plaques. But for many decades, these cells were considered just as merely bystanders or even secondary to neuronal damage. In the last 15 years, the role of neuroinflammation in AD has gained increasing attention. And this was mostly due to GWAS discoveries, which revealed that near 25% of AD risk loci were related with immune genes, such as TREM2. This left a shift from a neurocentric-based model toward a more integrative perspective. Here in Spain, we have a proud legacy in neuroscience. In 1891, Santiago Ramón y Cajal described neurons in great detail. He also illustrated a third element apart from neurons and oligodendrocytes. Here you can see one of his beautiful drawings of an astrocyte. Later, in 1919, a disciple of Santiago Ramón y Cajal, Pío del Río Ortega, used a new histochemical technique and was the first to describe the microglia. So, more than 100 years ago, two Spanish neuroscientists said that glial cells were more than just support cells. Today, we do know that microglia and astrocytes are key players in the immune response of the brain. Microglia are cells from a myeloid origin. They are considered the resident macrophages of the brain. The astrocytes have many homeostatic functions, such as maintaining the blood-brain barrier, contributing to synapses, and neurotransmitter clearance. So, in physiological conditions, both cells are neuroprotective. But under pathological conditions, microglia and astrocytes become reactive, showing different morphological, functional, and transcriptional changes. These initially try to contain or resolve the injury, but if the pathological stimulus persists, then chronic neuroinflammation leads to synaptic loss and neuronal death. So they are, in fact, two sides of the same coin. Depending on the context and the disease stage, they can either have a protective or a detrimental potential. But why do glial cells become activated in AD? Well, some bacteria like E. coli, Salmonella typhimurium, or S. aureus express surface proteins with amyloid-like structure. Microglia express pattern recognition receptors, or PRR, that can detect amyloid proteins in order to fight bacterial infections. In consequence, amyloid beta oligomers, due to their similar structure to bacterial surface, can stimulate these receptors and can be internalized by microglia. This triggers intracellular signaling cascades that promotes the release of pro-inflammatory cytokines such as IL-1 and TNF-alpha and the assembling of the inflammasome. The inflammasome then activates caspases and releases ASC specs. These ASC specs seem to be important. In vitro and animal models demonstrate that ASC specs can seed amyloid beta in Alzheimer's disease. Here on the left, you can see these ASC specs in red in the core of an amyloid plaque of mouse brain. And here on the right, in the core of an amyloid plaque of a human brain. Besides, the activated inflammasome can drive tau pathology. Here on the left, you can see a tau mouse model expressing the inflammasome, showing tau deposits in the hippocampus. And on the right, the same tau mouse model without the inflammasome and much less tau. And what about the astrocytes? The astrocytes also express receptors that can be activated by misfolded proteins like A-beta, and they can also become reactive in response to activated microglia. Reactive astrocytes can release pro-inflammatory cytokines that are harmful for neurons and also promote A-beta production and the spread of tau pathology. So, both microglia and astrocytes can increase neurodegeneration through multiple mechanisms and also influence amyloid and tau pathology. We now know that pathological AD changes can start 20 to 30 years before the first clinical symptoms. But when does neuroinflammation start? Since the glia react to misfolded proteins, it's logical to think that inflammation could be present in the earliest asymptomatic stages of AD. In fact, animal models show that focal glial activation may even precede the formation of amyloid plaques. Some researchers propose that neuroinflammation follows a non-linear, non-asymptotic, or non-exponential trajectory over time in AD. Instead, it may occur in multiple waves, the so-called multiple wave model. Each of these waves can be triggered by different factors, such as cell senescence, A-beta and tau oligomers, or the release of endogenous DAMPs from damaged cells. And let's move now to another important topic. Astrocyte and microglia reactivity is not a binary phenomenon. It's not as simple as saying active resting cells or A1, A2, M1, M2, according to the old classification. In this transcriptomic study, the authors demonstrate 7 different phenotypes of astrocytes, each one of them expressing different set of genes and associated with different brain regions in patients with Alzheimer's disease. Microglia are also very diverse. Transcriptomic studies confirm that there are multiple functional states which vary depending on the disease stage and the local environment. And TREM2 expression also varies among different microglial subtypes. A recent mouse study showed that high TREM2 microglia that usually is located near amyloid plaques seem to be functionally exhausted and showed a reduced response to TREM2 therapies. And if it is confirmed by further studies, this may explain the limited efficacy of TREM2 agonists at late disease stages. And how can we measure glial reactivity in vivo? Well, we now have robust biomarkers. Soluble TREM2, or STREM2, reflects microglial reactivity. For astrocytes, we have CSF and plasma GFAP, CSF YKL40 and CSF S100 beta. Interestingly, plasma GFAP seemed to perform better than CSF GFAP. These fluid biomarkers are useful to inform about general brain changes in inflammation, but they cannot tell us where the inflammation is happening. For that, we need imaging biomarkers. There are several PET radiotracers that have been developed so far. TSPO is the most studied one for microglia. We already have 3 generations of TSPO tracers. And for astrocytes, we have MAO-B tracers and I2BS tracers, among others. I have to say that none of them are fully specific for each glial cell, and in my opinion, they are still far to be used in clinical routine. So we need more precise and more dynamic imaging biomarkers. And please, now let me share with you the key findings of this study we have published last month in Alzheimer's and Dementia. In this work, we assessed changes in both microglial and astrocytic biomarkers in 211 cognitively unimpaired individuals from the Valdecilla cohort. The Valdecilla cohort is a prospective longitudinal study that started in 2018 and includes healthy volunteers of 55 years or older. We performed a collection of plasma, DNA, hair, and CSF from all participants. We also do 3 Teslas MRI, FDG PET, and a full neuropsychological assessment, and we follow individuals over time. To date, near 50% of the participants have completed the 5-year visit, but I should mention that the present work is focused only on baseline data. It's a cross-sectional study. CSF-AD biomarkers were measured using the LumiPulse, CSF S2, YKL40, S100 beta, and neurogranin were quantified using milliplex on the Luminex platform in our immunology lab, and CSF and plasma GFAP were measured using Simoa through a collaboration with the University of Gothenburg in Sweden. When comparing AT groups, we found that plasma GFAP and CSF S2 were increased in preclinical AD independently of age, sex, and ApoE4 status. We performed linear regression analysis adjusting for covariates, and we found that plasma GFAP was the only glial biomarker associated with lower A-beta ratio. All glial biomarkers correlated with p-tau. All except GFAP were associated with higher t-tau or NFL and neurogranin, and none of them were associated with cognitive performance by the PAC-5 score. We then used structural equation modeling to simultaneously assess all the direct and indirect relationships between glial biomarkers and AD pathological biomarkers. You can see the path model in this graph. Plasma GFAP showed an inverse bidirectional relationship with CSF A-beta ratio. CSF S2 directly influenced p-tau and neurogranin and correlated with CSF S100 beta. CSF YKL40 partially mediated the association between p-tau and t-tau, and finally, S100 beta and neurofilament light showed mutual influence. So in line with prior studies, our findings support that microglial and astrocyte reactivity occur early and impact the amyloid cascade in preclinical AD. Specifically, GFAP influences amyloid accumulation. S2 promotes tau pathology, and why KEL40 and S100 beta contribute to downstream neurodegenerative changes. And so far, we have been talking about the involvement of glial cells in Alzheimer's disease, but can we target them by therapies? To date, there are several TREM2 agonists that have been developed. The first one, AL002, was tested in a phase 2 clinical trial, but it failed to show clinical or biomarker benefit. However, it was associated with high rates of ARIA, for reasons not totally explained. The other two TREM2 agonists, one antibody and one small molecule, have showed safety and target engagement in early trials, but clinical efficacy is still unknown. We have to wait for phase 2 clinical trials, which are thought to start early. And as far as I know, no astrocyte-targeted therapies have been tested in clinical trials in humans in Alzheimer's disease. There are other approaches in preclinical research, such as inflammasome inhibitors or ASC-SPECS modulators. And we have been talking about neuroinflammation inside the brain, but the brain is not an immune-isolated organ. It's connected with the periphery. In AD, neutrophils, macrophages, and T lymphocytes can infiltrate the brain. Mouse models of AD show that microglia-mediated T-cell infiltration promotes neurodegeneration, and that systemic inflammation reduces the clearance of A-beta by the microglia. And there is also a huge body of evidence from epidemiological studies supporting that chronic inflammation in midlife is associated with an increased risk of dementia. This leads to an important question. Can we reduce the risk of AD by treating all these factors that promote a pro-inflammatory state? At present, there are 25 clinical trials in Alzheimer's disease based on the modulation of inflammatory or immune-based mechanisms. So targeting inflammation is, in fact, a very active and promising field of research. We are near to end this presentation, but there are still many open questions. First, neuroinflammation in AD may follow a multiple wave course, so different brain regions potentially can be at different disease stages at the same time. So we need longitudinal studies combining fluid and imaging biomarkers. Second, microglia and astrocytes can have multiple phenotypes, but we only have biomarkers for a few of them, so we need new biomarkers to capture the full complexity of these cells. And finally, the brain is not an isolated organ. It's connected with the periphery, but its exact role in AD pathogenesis is still unclear. In conclusion, glial cells are highly dynamic and heterogeneous, with both protective and harmful potential depending on the disease stage and local environment. Chronic glial response impact the amyloid cascade events from early preclinical stages of AD. TREM2 modulation may be a potential therapeutic target in AD, but we need more information, more knowledge about its biology. And finally, understanding and targeting neuroinflammation offers a promising path to changing the trajectory of Alzheimer's disease. Thank you very much for your attention. And now I would like to invite the next speaker, Dr. Estrella Morenas, neurologist at Hospital 12 de Octubre de Madrid, who is going to talk deeper about the therapeutic potential of TREM2 modulation. Thank you.
Thank you, Marta, for the presentation. And thanks to the organizer for giving me the opportunity to give this talk today. And here I take the point that Marta finished with the potential of the TREM2 modulation in Alzheimer's disease as a disease-modifying therapy. So here in this first slide, I want to highlight the temporal line since the first definition, since the first description of TREM2 as a risk factor for Alzheimer's disease. Here I think this is the fourth time that we see this graph. So here you have TREM2 as a risk factor for Alzheimer's disease, and since 2013 there have passed only 10 years until we have had the first clinical trials ongoing trying exploring the therapeutic potential of the modulation or the activation of microglia modulating TREM2 functions. Unfortunately, as Marta has mentioned, just at the end of 2024, we had the first clinical trial in phase 2 that have failed. So here, this is not a spoiler of this presentation, but I think it's important to have this in mind to reconsider all the data that we have, again, to rethink the TREM2 role as a therapeutic target in Alzheimer's disease, knowing all the biomarker approaches that we have, the knowledge that we have from biomarker approaches up to date. And the third thing that we have to keep in mind when we speak about TREM2 is this complex biology of the TREM2, because this is not only about the A-beta deposition, TREM2, and the activation of microglia by TREM2 have different functions, different roles as lipid metabolism, proliferation and chemotaxis, and the role in the synapses. That is everything interconnected, and of course, it is the image of the different ways of activating microglia originating different phenotypes. That is, so it builds up a very complex biology, but that we need to understand. So, if we center in Alzheimer's disease and starting by the beginning, so the role of TREM2 functions on Alzheimer's disease pathology, we know from animal models that TREM2 activated by activating microglia has a very important role in the A-beta pathology and modulation the A-beta pathology, limiting the first A-beta aggregates, compacting the A-beta plaques by its working together with ApoE, and here is the more complex role of TREM2-dependent activated microglia. There is the effect on tau pathology. So here, based on animal models, we have studies that show a beneficial effect of activated TREM2, activating microglia via TREM2 on tau pathology, tau pathology, but we have also other studies, other animal model studies that show that under chronic activation of TREM2, we may have a detrimental effect on AD or A-beta-dependent tau pathology. So, with that in mind, here TREM2 has a very huge advantage, that is that we can measure, so TREM2 is a membrane protein that is located in the membrane in microglial cells, and it is processed in the cell membrane and generates the cleaved-soluble TREM2 fragment that can be detectable in CSF, and then we can use it as a biomarker of TREM2-dependent microglial response. And that, so it makes it easier for us to go from disease model to study its role in clinical cohorts. But it's also a disadvantage because maybe it has made us to run too much into clinical trials or into clinical cohorts before the understanding of all the complexity of the biological roles of TREM2-dependent microglial activation. And the first that we know about soluble TREM2 in clinical cohorts, that is, it is elevated both in sporadic Alzheimer's disease and in autosomal dominant Alzheimer's disease in early symptomatic stages. But what is triggering TREM2 until arriving to this maximum of soluble TREM2 in CSF? So that was what we look at in the DIAN cohort, that, as you know, is a cohort of genetic Alzheimer's disease, in which there are included mutation carriers that are going to develop in the disease, and also the relatives unaffected by the mutation, and in which we can study very nicely what is happening in this preclinical phase of the disease. So what we saw was that soluble TREM2 started to increase even 21 years before the expected symptom onset. It's a time point that is very near the start of the accumulation of A-beta in the cortex as measured by A-beta PET. And what we also saw in this cohort is that this increase of soluble TREM2, this longitudinal increase in the soluble TREM2, was very related with lower levels of CSF A-beta 42 in the CSF, but it was not related with a higher uptake in the amyloid PET, which led us to hypothesize that one of the main triggers of the increase of soluble TREM2, or what is the same, the activation, the TREM2-dependent activation of microglia, was the initial oligomerization or the initial A-beta pathology when it is still soluble, and not the cortical deposits of the formed amyloid plaques. And what we also saw, reinforcing this relationship between the TREM2-driven microglial activation and A-beta pathology, was that in a disease phase-dependent manner, higher longitudinal soluble TREM2 increase was associated with a slower amyloid deposition. And here we can see that in the preclinical, in the presymptomatic mutation carriers, a higher longitudinal increase in soluble TREM2 was related with a slower longitudinal decrease in CSF A-beta 42, while we could see a slowing of the increase in the uptake in the amyloid PET related with a larger increase in soluble TREM2, representing this microglial activation, specifically in symptomatic. So here we are having a phase-dependent effect on the different phases of the amyloid pathology. And at a longitudinal level in this study, we didn't find any relationship between the longitudinal dynamics of soluble TREM2 and tau markers. And also, higher soluble TREM2 increases are associated with better neuroimaging outcomes. So supporting this protective role here, what we can see, also in a phase-dependent manner, is that in genetic Alzheimer's disease, in preclinical mutation carriers, a higher longitudinal increase of soluble TREM2 was related with a slower shrinkage, cortical shrinkage in the precuneus, specifically in the precuneus, while in sporadic AD, we could also see that higher baseline soluble TREM2 levels in an early symptomatic phase was related with a better, slower hippocampal shrinkage. So here we have the phase-dependent effect and the region-specific effect dependent on if it is genetic or sporadic Alzheimer's disease. And very importantly, what we saw was that higher longitudinal soluble TREM2 increase in presymptomatic Alzheimer disease, in presymptomatic genetic Alzheimer disease, what very strongly associated with a slower cognitive decline. And that here I'm also highlighting this phase-dependent effect, because we very nicely could see here the correlation between a higher longitudinal increase in soluble TREM2, representing again the microglial activation with the relationship with this a slower cognitive decline, but only in the pre-symptomatic mutation carriers before the symptom onset. And here, and we wouldn't see this relationship when symptoms have started. And here in this graph, what we could see is that if we stratify patients by its soluble TREM2 levels, those participants or mutation carriers that had higher soluble TREM2 levels had a more stable cognitive evolution. And that, of course, so it shows that the TREM2-dependent response in early phases of AD, specifically in preclinical, pre-symptomatic phases, has a protective effect, or it suggests that. And it, of course, have clinical implications for treatment and for the design of clinical trials. And besides the genetic AD, it has been shown too that higher levels of soluble TREM2 are related to a slower cognitive decline evolution in sporadic Alzheimer's disease and age. So here we have the early symptomatic AD, and here we can see as higher soluble TREM2 levels at baseline are related with a slower cognitive decline in the acne cohort, in the biofinder cohort, and in cognitively unimpaired individuals in the alpha cohort. And here we have a very interesting thing, that is that the baseline levels of soluble TREM2 have an effect on cognition that is dependent on the A-beta status, which reinforces these phase-dependent effects. And then, if we have this nice clinical data from different cohorts with different ethnicities, with different biological backgrounds, so highlighted by different groups with different methods, why TREM2 therapies are failing. Of course, I don't have the answer. I want to highlight different possible key points that we have to keep in mind to the clinical trial design and to the design of further studies from a basic research perspective. So here the first key point is the controversial relationship between TREM2 and tau, because we have some animal model-based studies that argue that TREM2 activation of microglia has a protective effect on tau related with A-beta. And we have also clinical data showing that those patients or those participants with higher soluble TREM2 levels have better longitudinal trajectories in the accumulation of tau pathology. And this is, I think, from the biofinder. And here we have another study in which they saw that soluble TREM2, so those participants with higher levels of soluble TREM2 have, or those participants with lower levels of soluble TREM2 had a higher increase in the tau PET uptake related to the A-beta 42, A-beta ratio. But of course, there are other studies, so from a basic research perspective, that, and in fact, this one I like very much, and this one is very interesting, that argue for a detrimental effect of the TREM2 driven activation on tau pathology. Regarding clinical studies supporting this hypothesis that is detrimental, we have a lot of cross-sectional associations between soluble TREM2 and tau pathology, but cross-sectional associations indicated the cross-sectional association between those markers as if they are both a response of, for example, A-beta pathology. You can think that they are going to increase together, so they are correlating cross-sectionally. Longitudinally, there are some studies that have also so a mediation effect of solvent TREM2 between the amyloid, between amyloid pathology to tau pathology. And also, very interestingly, there have other studies that are not centered in TREM2, but have also pointed to a potential role of microglia in the spreading of tau pathology. And what we saw in the Diane cohort? So we saw, again, a phase-dependent effect, so specifically in preclinical mutation carriers, we saw that those with a lower soluble TREM2 levels had a relationship between higher increase in the amyloid PET related with the higher increase in CSF phospho-tau. But we couldn't see this relationship in mutation carriers with high levels of soluble TREM2, and for that we hypothesized that TREM2 might have a protective effect also on A-beta-dependent tau pathology. And with all together, so with all this data, we proposed in 2022 a therapeutical scheme in which we proposed this therapeutic window for TREM2-focused treatment encompassing all the preclinical stages and the MCI. With the failure of the phase 2 clinical trials in an MCI state, and with all the data, and of course, taking into consideration that we don't know exactly what TREM2 activated microglia is directing in tau pathology, we can rethink this therapeutic window for TREM2 tighten to the preclinical stage. This is pretty difficult to design in clinical trials, and also taking into consideration different biomarker profiles, specifically tau positive or tau positivity or tau negativity in CSF or in PET with PET tracers, and also maybe the levels of TREM2 in CSF to be able to stratify patients in those who are able to increase the TREM2 response, or they are not able to increase the TREM2 response because the TREM2 response is already very high. A key point too is, again, the complex biology of TREM2. I have already mentioned this. We have focused on the relationship between TREM2 activation and A-beta deposition, but there are many others aspects that we have to deepen into it, and we have to know or understand how they are related all together. And here, briefly, I'm presenting some, not preliminary, but exploratory data of my starting group on the relationship between soluble TREM2 and the synaptic dysfunction. And what we did was to go to the Wisconsin cohort of healthy aging volunteers, and we studied the relationship between different few synaptic markers and the longitudinal increase or dynamics of soluble TREM2. And what we saw was that both lower levels of neurogranin and lower levels of alpha-synuclein were related with a higher longitudinal increase of soluble TREM2. What made us to hypothesize that in absence of A-beta pathology, the synaptic dysfunction may act as a trigger of the TREM2 activation. And as we only had measured only 2 markers, we also explored the relationship with S100B, which is an astroglial marker, that signaled the response to synaptic damage. And what we found was that higher levels of S100B were also related with higher longitudinal increase of soluble TREM2, while we didn't find any relationship with the GFAP in CSF. That makes us to endorse this notion that the synaptic dysfunction in absence of A-beta pathology is a trigger of TREM2 activation. And we also saw that higher soluble TREM2 at the baseline were related with more stable levels over time in neurogranin and have a trend towards an association with more stable levels in alpha-synuclein. And we also check in another cohort this relationship, and we could see that we found, and we replicate the same association, but in the alpha plus cohort, only in the subset of patients in which they have normal biomarker profile or a biomarker profile that is not suggestive of an underlying AD neuropathological process. And going going further in relationship with this relationship between soluble TREM2 and synaptic marker, we also explore soluble TREM2 in a cohort of DLB patients. That is characterized, DLB, as you know, by a huge synaptic dysfunction with less neuropathology. And here, what we saw was that soluble TREM2 levels were increased in prodromal DLB, also in DLB, but especially in prodromal DLB, where there is still not overt neurodegeneration or neuronal death, but again, a huge synaptic dysfunction. We also here, we wanted to check if this increase was due to an Alzheimer's copathology, as we have seen that the initial this metabolism of A-beta was a main trigger of the increase of soluble TREM2, and what we saw was that soluble TREM2 levels were higher in those patients without a biomarker profile suggestive of amyloid accumulation than in those with a biomarker profile of amyloid accumulation. And related to the tau status, we didn't find any difference between tau plus and tau minus prodromal DLB patients. And what we saw also very nicely was that higher soluble TREM2 levels during the prodromal stage were related with a slower cognitive evolution in prodromal DLB, but not in DLB dementia. What again reinforces this phase-dependent effect independent of AD copathology in this case, and point to a potential utility of the TREM2 modulation therapies in other neurodegenerative conditions. And the key point 3, and this is the last point, is the biological role of soluble TREM2. So here we have that TREM2 is processed in the cell membrane, generated this clear soluble TREM2, and we use that as a biomarker of the microglial response driven by the TREM2 activation. But we have two other isoforms that are directly soluble in which we don't know its function. And it's not trivial that they are there because this is the co-occurrence of these three isoforms is happening only in humans. And they should have a clinical or a biological meaning. And reinforces this biological role of soluble TREM2, here we have different polymorphins protective for AD that directly modulate the increase in CSF of soluble TREM2. So, supporting this biological effect of these forms. And my take-home message here is that we have a robust biomarker evidence that supports the therapeutic potential of TREM2 modulation. We have data, so very robust in different cohorts in genetic AD and in late onset AD, and we have some data in other age-related conditions. But we need to bridge these mechanismic and conceptual gaps that we have for the successful of the TREM2 modulation. So it's crucial that we understand the complexity of the biological function of the TREM2 activating microglia, the relationship with other cell types as astrocytes, and the regulators of soluble TREM2 forms and the TREM2 activation in microglia. Very important to understand the biological role of soluble TREM2 isoform, as many therapies reduce or stabilize this soluble TREM2 isoform. So we have to know what is happening, so what they are doing to know if this negative effect is because we are reducing soluble TREM2, because it seems that soluble TREM2 itself might have a beneficial role. And very important, we have all the data shows a context-dependent effects. And we cannot oversee that because it has direct impact on clinical trial design. And not accounting for that may lead us to have bad clinical design that have a result of the failure of the clinical trials that we are trying. And with that, again, thanks to my group and all of you for your attention.
Thank you very much, Estrella. The next talk will be by Dr. Amaia Arranz from Achucarro Basque Center for Neuroscience, who is going to talk about divergent effects of human astrocytes with different ApoE variants on key AD hallmarks.
Good afternoon, good evening, everybody. First of all, I would like to thank the organizers of this meeting for inviting me to give this seminar. Today, I will explain you a project that we are running in the lab in which we generated a chimeric mouse model of Alzheimer's disease with human astrocytes carrying either ApoE3 or ApoE4 variants, and we found that these human astrocytes with different ApoE variants have divergent effects on key Alzheimer's disease hallmarks in these chimeric mice. So at this point of the talk, I'm sure you don't need an introduction about Alzheimer's disease. So I will just briefly mention that Alzheimer's disease is the most common chronic neurodegenerative disease, and the most common cause of dementia in the elderly. And that Alois Alzheimer described already more than a century ago the main hallmarks of the disease, which are the following: the extracellular accumulation of the amyloid beta peptide that aggregates and forms the amyloid beta plaques, the abnormal hyperphosphorylation of the tau protein that acquires a conformational change, pathological change, giving rise to the neurofibrillary tangles, the inflammatory response with both astrogliosis and microgliosis, and an increased number of both astrocytes and microglia surrounding the amyloid beta plaques. And there is also synaptic dysfunction and neuronal loss, leading to brain atrophy, cognitive deficits, and dementia. And I would like to stress the fact that although we know for a very long time these hallmarks, and they have been very well studied and characterized, we still don't completely understand the mechanisms underlying AD pathogenesis. And drug development is so far yielding very limited results, underscoring the need to find new pathways and new drug targets for this terrible disease. How is it that we cannot respond to Alzheimer's disease? Well, a main reason is that most of what we know about the brain comes from studies in animals, typically rodents. And while we have learned a lot from these rodent models of the disease, the characteristics, or some of the characteristics that make a human brain unique and uniquely susceptible to neurodegenerative diseases such as Alzheimer remain mysterious, highlighting again the need of human-based models that allow capturing the complexity of the disease process. Besides, AD research has remained neurocentric for a very long time, but recent studies, US and other studies, are highlighting the importance that glial cells play in Alzheimer's disease, as most of the genes associated with increasing the risk for developing late-onset Alzheimer's disease are mainly expressed by these glial cells, either microglia, oligodendrocytes, or astrocytes. Among these genes, ApoE4 is a major genetic risk factor for developing late-onset Alzheimer's disease. And ApoE is mainly expressed by astrocytes in homeostatic conditions, and microglia also express ApoE in pathological conditions. Well, you have heard this morning a lot about ApoE, so I will briefly mention that is an apolipoprotein belonging to a family of fat-binding proteins, and its main function is transporting cholesterol and lipids to neurons via ApoE receptors. You have also heard that there are in humans, it's polymorphic, and there are three isoforms. ApoE4, which is the major risk factor for developing Alzheimer's disease, ApoE2, which is the protective variant, and ApoE3, which is the most common among the population and is considered the neutral variant. And these isoforms just differ in 2 amino acids that impacts the protein structure and function. And in Alzheimer's disease, we know that ApoE is binding to the amyloid beta plaques, and it's influencing its clearance, aggregation, and deposition. And it's also affecting the inflammatory responses led by astrocytes and microglia. Well, coming back to the role glial cells play in Alzheimer's disease pathogenesis, now the field of Alzheimer's research is focusing mainly on understanding and studying the role microglia is playing on Alzheimer's disease. But cells such as astrocytes are still under investigated in Alzheimer's disease. Well, I find this is remarkable as astrocytes are integral components of the central nervous system, essential for maintaining homeostasis and proper brain function, and they are also affected in Alzheimer's disease. So in this project, our goal was to study how astrocytes contribute to AD progression and to understand the influence of different ApoE variants on this process. But we know that major differences exist between mouse and human astrocytes at molecular level, at functional level, and at morphological level. And as this is also happening not only with astrocytes, but also with other brain cells, we pioneered a few years ago, when I was postdoc in the lab of Bart Struber, an approach to study these human brain cells in vivo in the context of Alzheimer's disease. We generated the first humanized chimeric models of Alzheimer's disease by transplanting human iPS-derived glia and neurons into the brain of AD mouse models. Importantly, this powerful technology allows us to study human cells, human brain cells, in the live brain exposed to amyloid beta and other AD-associated pathologies. By using these humanized chimeric models of Alzheimer's disease, we were able to reveal a selective vulnerability of human neurons to AD-associated pathologies not present in the mouse neurons. Also, divergent responses of mouse versus human glial cells to major AD hallmarks, mechanisms leading to the death of human neurons, and also potential therapeutic strategies, highlighting the importance of studying human brain cells in Alzheimer's disease. Well, to generate the chimeras with human astrocytes and to be able to study the contribution of these human astrocytes with different ApoE variants in vivo in the chimeras, we developed the following approach. We first generated isogenic iPS lines from 2 Alzheimer's disease patients carrying either ApoE4 or ApoE3 alleles. And we differentiated these iPS cells in vitro to obtain astrocyte progenitors that we were injecting into the brain of newborn AD mouse models or wild-type litter mates that were also immune deficient. We let the mice age for 5 to 6 months when the major Alzheimer's disease hallmarks are already developed in these animals, and we use these mice to understand how these human astrocytes with different ApoE variants were contributing to main Alzheimer's disease pathological features. Well, we found that these transplanted cells were integrating efficiently in the mouse host brain. In some of these cells, as you can see here, were integrated individually, but in most of the animals analyzed, in fact, in almost 80% of the animals analyzed, the majority of the cells were integrated in one cortical hemisphere, and so the morphological features of interlaminar astrocytes that are present in layer 1 of the human cortex. We also found that these transplanted cells that were tagged with TD tomato and can be visualized with an RFP antibody were also expressing human-specific markers, such as human nuclei. And they were showing astroglia identity. So they were expressing my markers of astrocytes, such as GFAP, vimentin, aquaporin and S100 beta, and there was no or minimal expression of markers of other cell types, such as oligodendrocytes or neurons. And these cells, these transplanted cells, were also contacting blood vessels, as you can see on this image. And both ApoE3 and ApoE4 human astrocyte progenitors were similarly differentiated into astrocytes in vivo in the chimeric brains. Well, they were also, these human astrocytes, showing functional integration. They were able to contact blood vessels with their ENFIT, as human astrocytes are doing in our brains, and they were also able to establish gap junction connections. They were expressing the gap junction marker connexin 43, and when we loaded these cells, these human astrocytes, with a dye, in this case the Alexa 488, we found the presence of the dye in neighbor mouse host cells. So this dye presumably reached to these cells through gap junction connections. These human astrocytes, they were also showing electrophysiological properties very similar to astrocytes in our brains. Well, as I told you before, astrocytes are the cells that are mainly expressing and producing ApoE. So we wonder if these human astrocytes were also expressing ApoE in vivo when they were transplanted in these mice. And the answer is that they do. We measured the levels of human ApoE by ELISA, and we found that in the chimeras, in brain homogeneids from the chimeras with ApoE3 human astrocytes, the levels of ApoE, of both soluble, insoluble, and total ApoE, were much higher than in the chimeras with ApoE4 astrocytes. We also analyzed the expression of human ApoE in these human astrocytes by immunofluorescence, and we found that in the ApoE3 astrocytes, the levels of ApoE were much higher than in ApoE4 human astrocytes, both in wild-type animals and also in the Alzheimer's disease mouse model. And while the levels of ApoE seem to be upregulated in ApoE3 human astrocytes in Alzheimer's disease mice compared to wild-type mice, the levels of ApoE in ApoE4 astrocytes don't seem to be significantly modified between wild-type and AD animals. We were also interested in knowing if these human astrocytes were also able to secrete ApoE to the extracellular space, and the answer is that they do. And also, again, we measured the levels of ApoE in an area of 100 micrometers from the somas with the human astrocytes, and we found increased levels of human ApoE in regions close to ApoE3 astrocytes than in regions close to ApoE4 astrocytes. And very interesting, this ApoE is able to bind to the amyloid beta plaques in the chimeric brains. And we found that both ApoE that was secreted to the extracellular space and ApoE that was from the astrocyte processes that were in direct contact with the amyloid beta plaques was binding to the plaques. And we found here very similar levels. We measured the levels of ApoE in these amyloid plaques that were very close to the human astrocytes, and we found similar levels of ApoE in plaques close to ApoE3 and in plaques close to ApoE4 astrocytes. So this is the summary of this first part of my talk. We were able to generate a chimeric model of Alzheimer's disease with human astrocytes that were able to integrate efficiently in the mouse host brain and show morphological features of human interlaminar astrocytes. These human astrocytes are able to integrate functionally, and they are expressing and secreting human ApoE, with differences between ApoE3 and ApoE4 human astrocytes. And the ApoE, this human ApoE, is able to bind to the amyloid beta plaques. So at this point, once we have the chimeric model well established, we focus on understanding how these human astrocytes were contributing to AD progression. And the nice thing of this model is that the way human astrocytes integrate in the mouse brain allow us to study their contribution to pathology in the same region, well, in the same animal and in the same region of the brain, in this case, in the cortex. Oops. because we can compare the cortical hemisphere that has the human astrocytes with the cortical hemisphere, the contralateral cortical hemisphere that has no astrocytes. We analyzed this way how human astrocytes were contributing to AD, to amyloid beta pathology, in this case, using immunofluorescence and staining the plaques with theoflavin, and we found that in these regions with ApoE3 astrocytes, amyloid beta burden decreased compared to contralateral regions. There were a less number of plaques per area, the size of the plaques was smaller, and the area occupied by the plaques is also smaller. However, in areas with ApoE4 astrocytes, there were more plaques per area compared to the contralateral sites. We also validated these results using another antibody, 63, to stain total amyloid, and we found very similar results. In areas with ApoE3 astrocytes, amyloid burden decreased compared to contralateral regions. But in areas with ApoE4 astrocytes, we found more plaques per area compared to the contralateral sites. We also measured the levels of both soluble and insoluble amyloid beta by ELISA and found that, again, in areas containing these ApoE3 astrocytes, the levels of both insoluble and soluble amyloid beta decreased compared to the contralateral sites. However, we were not able to find differences in the levels of both soluble and insoluble amyloid beta in ApoE4 chimeras. In addition to measuring amyloid beta load in these regions with the human astrocytes, we also measure this amyloid beta burden in areas that are just very close to the ApoE3 or ApoE4 astrocytes. And we found a similar trend. So in areas that are close to the ApoE3 astrocytes, you can see the processes of the astrocytes here, amyloid beta burden decreased compared to the contralateral regions. However, we were not finding differences in amyloid beta burden in regions close to ApoE4 astrocytes. We also analyzed if these human astrocytes have an effect on plaque morphology or compactness, and the answer is that they have an effect, and in fact, these plaques that are close to ApoE3 astrocytes are more compact and more circular than in the contralateral side. However, there are no effects in the plaque morphology or in the compactness of the plaques in plaques that are close to ApoE4 astrocytes compared to the contralateral sites. Well, overall, this data is telling us that there is a contribution of human astrocytes and the human ApoE that they secrete to the extracellular space to amyloid beta pathology in vivo in these chimeras, and that there is a distinct impact of human astrocytes with different ApoE variants. We also wonder if there was a crosstalk between these human astrocytes that we transplanted and the mouse microglia. And for that, we've studied plaque-associated microglia, staining these microglia cells with IVA1. And we found that in areas close to ApoE3 astrocytes, there was a reduced number of microglia cells surrounding the plaques compared to the contralateral sites. In contrast, in areas close to ApoE4 astrocytes, there was an increased number of microglia surrounding the plaques compared to the contralateral sites. We saw very similar effect on total microglia when we analyzed total microglia, not only the plaque-associated microglia. And as you can see on these images, in areas that are close to these ApoE3 astrocytes, total microglia number decreased compared to the contralateral sites. However, we were not finding any differences in total microglia in regions close to ApoE4 astrocytes. So this data is suggesting that these ApoE3 astrocytes, or the ApoE they secrete to the extracellular space, on any other factors they are secreting, are inducing a more homeostatic state on microglia. And the opposite is happening in the case of ApoE4 astrocytes. So to validate this idea, we analyzed the expression of P2Y12, which is a marker of homeostatic microglia. And we found that in these areas that are next to ApoE3 astrocytes, P2Y12 expression around plaques is higher than in contralateral areas. However, in areas close to APOE4 astrocytes, P2Y12 expression doesn't change compared to the contralateral regions. We also analyzed the expression of CLEC7A, which is a marker of disease-associated microglia, and we found that the levels of CLEC7A surrounding the plaques were down in areas close to APOE3 astrocytes. However, the levels of CLEC7A were up in areas close to APOE4 astrocytes compared to the respective contralateral sites. To assess some potential functional effects of these microglia responses, we also analyzed the coverage of the plaques by these microglia cells. And we found that although there is less number of microglia surrounding the plaques in areas close to APOE3 astrocytes, and an increased number of microglia in these regions close to APOE4 astrocytes, plaque coverage by microglia didn't change in these regions compared to contralateral regions. That means that probably this microglia close to APOE3 astrocytes is more efficient covering the plaques. However, this microglia in regions close to APOE4 astrocytes is less efficient, and there is a need of more microglia to have the same levels of plaque coverage. So, well, overall, these results suggest that there is really a crosstalk between the human astrocytes and the mouse microglia, and that these human astrocytes are modulating the responses of the microglia around plaques with differences between different astrocytes with different APOE variants. We also analyzed how these astrocytes were contributing to tau pathology, and for that we use 88, which is a marker of phosphorylated tau, hyperphosphorylated tau, and we analyzed tau, 88 positive puntas surrounding the plaques, the amyloid beta plaques. And we found that in areas that are next to APOE3 astrocytes, there is a decreased number of 88 positive punta, and in contrast, in areas close to APOE4 astrocytes, there is an increased number of 88 positive punta compared to contralateral regions. This is an overview showing the same. So basically, in these regions that are close to APOE3 astrocytes, there is less number of tau puntas surrounding the plaques, and in areas close to APOE4 astrocytes, there is an increased number of 88 positive puntas surrounding the plaques. And finally, we also analyzed neuritic dystrophy, which is one of the first signs of neurodegeneration in Alzheimer's disease. We stained the dystrophic neurites with APP, which is highly expressed in these dystrophic neurites, and we found that they were smaller in regions close to APOE3 astrocyte, and they are bigger, these dystrophic neurites surrounding the amyloid beta plaques, in areas close to APOE4 astrocytes, comparing with their contralateral sites. This is an overview showing the same results. And well, this is the final conclusion and the summary of this second part of my talk. Well, we found that these isogenic APOE3 and APOE4 human astrocytes have a differential impact on main Alzheimer's disease hallmarks, such as amyloid beta pathology, tau pathology, microglial responses, and neuritic dystrophy. And while APOE3 human astrocytes are driving improvements, isogenic APOE4 human astrocytes are aggravating these pathologies, having a more impactful role on Alzheimer's disease and neurodegeneration than we previously thought. And as a final general conclusion of this work in chimeric animals is that these chimeras are providing new ways to study patient-derived cells. We can study not only human astrocytes, but other human neurons or other kind of human glial cells, derived from Alzheimer's disease patients, but also from patients with other kind of neurological or neurodegenerative diseases. And we can use these chimeras to understand how these human brain cells affect disease progression, as we did on this work, but we can also use these chimeras to understand how a disease brain environment is affecting the human cells. And I will finalize my talk by saying thank you to all the members of my lab, especially Joan and John, who made most of this work, and also our collaborators and all our funding agencies. And I will be happy to take some questions later.
Thank you very much. And now it's my pleasure to introduce Dr. Eduardo Zimmer from Universidade Federal Rio Grande do Sul, Brazil, who is going to talk about the cellular origins of neuroinflammation in Alzheimer's disease.
Muchas gracias por recibirme. Estoy muy contento de estar aquí hoy. And I would like to thank Pascual for inviting me and all the organizing committee. It's really a pleasure to be here. So these are my conflicts of interest, but the data that I'm going to show today is not related with any of these conflicts. Let's start with the basic concept. What does neuroinflammation mean in Alzheimer's disease? It's classical inflammation, but in the brain. This is a very nice perspective paper from Elena Glia, which was a very important glial biologist, a Spanish glial biologist that passed away two years ago, in which she proposed that we have been used the term neuroinflammation in a very non-specific way. But how can we measure neuroinflammation? We can use imaging biomarkers, and as Marta showed before, we have tracers for tracking astrocyte reactivity based on MALB overexpression or I2BS overexpression. And here you can see what happens in early stages of amyloid pathology. We have this increased signal using these tracers. But we can also measure microglial reactivity with imaging biomarkers, and we have a lot of data with TSPO tracers that also increase as a function of Alzheimer's disease. In fact, we believe that we have two waves of TSPO reactivity, an early wave and a late wave of TSPO increased signal. So we can measure neuroinflammation using these two modalities of tracers. We can also use fluid biomarkers. So this is what happens with GFAP, which is a classic marker of astrocytes. As you can see, it increases as disease progresses. We can use ICL40, that also has a similar pattern of increased levels as disease progresses. And for microglial reactivity, we can use SRAN2, which, as you can see here, increases also as disease progresses. So we have all these biomarkers that have been used to measure glial reactivity in the brain. So, in fact, based on the evidence that we have with biomarkers in living individuals, neuroinflammation means glial reactivity in Alzheimer's disease. And what we expect to see, as you can see here, they follow the disease stages. So with astrocytes, they sense the amyloid pathology, they change phenotype and sense tau pathology, and then they are different again in the neurodegeneration phase. So this is what we expect to see as disease progresses. But this is very important. It's the former concept. We first thought that we have like microglial and astrocytes in an homeostatic phenotype in the healthy brain, and then they become reactive and they change phenotype in response to amyloid and amyloid plaques and tau tangles. But this is the forming concept. Now we know that astrocytes, they assume multiple phenotypes in response to amyloid and tau pathology, which are dependent on brain region, which are dependent on disease stage. For example, we can have an astrocyte that's GFAP positive and other astrocytes that is YCL4 positive. We can have a microglial cell that is SRAN positive and other microglial cell that's TSPO positive. So by using these biomarkers, we are measuring different things in the context of all this glial heterogeneity. But where are the glial cells in the ATN framework, in the framework proposed by the Alzheimer's Association? They are here, as you can see, in a label that they use like inflammation or astrocyte activation, and the marker proposed is GFAP. So Juan Fortea was talking about nomenclature issues, and this is a nomenclature issue because astrocytes, they are active all the time. What we see in pathology is astrocyte reactivity. But I will try to convince you in the next slides that, in fact, these glial biomarkers, they are everywhere in the ATN system. And let's start with, of course, first we need to explain what GFAP in the blood means, which is the proposed biomarker. And this is a very nice paper from Pascual and Alberto, where they did the neuropathological validation of GFAP. So it means that GFAP levels in the blood are associated with GFAP immunoreactivity in the brain. I have the honor to comment this paper on brain. And as you can see, blood GFAP reflects astrocyte reactivity to Alzheimer's disease pathology. So let's see what happens with amyloid biomarkers. And this is the classic astrocyte reaction to amyloid plaques. We have an amyloid plaque in the middle, and we have these hypertrophic astrocytes staying with GFAP here in the vicinity of amyloid plaques. And together with Andrea Benedetto and Pedro Rosa Neto, we show that GFAP levels increase in early amyloid pathology. In cognitively unimpaired individuals that are positive to amyloid plaques, GFAP levels are already increased. And it keeps increasing as disease progresses. So let's add tau to the model, tau biomarkers. And this is what we expect when you see the association between amyloid PET and p-tau 181 in the blood. We expect to see this linear association. But if we divide these individuals in GFAP positive and GFAP negative, it's completely different. And together with Bruno Bellever and Tarek Pascual, we show that even if you have a lot of amyloid burden and you are GFAP negative, you are not going to have increased levels of p-tau in the blood. But if you are GFAP positive and have a high amyloid burden, we are going to have high levels of p-tau 181 in the blood. So it seems that to have the association between amyloid and tau, we need to have increased astrocyte reactivity. Okay, now we are back. So even if this is something that Marco De Bastiani did, which is a postdoctoral fellow in the lab, even if we sort only GFAP positive astrocytes in a model of amyloid and a model of tau, here he did this sorting astrocytes from APP-PS2 and a MAPT model to replicate how they interact, the responses that astrocytes have to different pathology. We call them amyloid and tau astrocytes. We can see that the amyloid astrocytes are completely different than the wild-type astrocytes, and the tau astrocytes are really different from the wild-type astrocytes. But when we try to compare them, we can see that they are completely different. They are GFAP positive astrocytes, but they share only 30 differentially expressed genes. We have more than 200 genes that are only expressed in the amyloid, only differentially expressed in amyloid astrocytes, and we have around 500 differentially expressed genes that are only expressed in the tau astrocytes. And the amyloid astrocytes are more related to neurotransmission and proteostasis, whereas the tau astrocytes were more related to inflammation and DNA RNA processing. So even GFAP positive are completely different in response to amyloid and tau pathology. So can we measure this astrocyte heterogeneity with biomarkers in Alzheimer's disease? And this is the work of Bruno Bellever first. She meta-analyzed more than data from more than 3,000 individuals, and she found that GFAP and YKL-40 are consistently increased in the CSF of Alzheimer's disease patients. What we did next with JP Ferrari Souza, we evaluated whether these astrocyte biomarkers are indicating different pathology in the Alzheimer's disease brain. And as you can see here, it seems that GFAP is more related GFAP is more related to amyloid, and YCAL40 is more related to tau pathology, which means that we have two different astrocyte biomarkers that are associated with different phases of Alzheimer's disease. So, what about neurodegeneration? What is the role of astrocytes in neurodegeneration? So, we already covered amyloid biomarkers, tau biomarkers. Let's talk about neurodegeneration right now. And this is the current concept of neurodegeneration, based on the Nature portfolio. So, neurodegeneration refers to the progressive atrophy and loss of function of neurons, which is present in neurodegenerative disease such as Alzheimer's disease. As you can see here, we have progressive atrophy and loss of functions, loss of function of neurons. We're not talking anymore about neuronal death in the concept. And how do we measure that based on the Alzheimer's Association criteria? We can use MRI for evaluating brain atrophy, or we can use FDG PET to evaluate brain glucose metabolism. So let me share with you three important informations about glucose metabolism in the brain. So this is the first one that I call location. As you can see here, and you all know, astrocytes, they have an end fit that is connected to the blood vessel, which has glucose transporters. So the first question here is why glucose needs to go to the extracellular medium, to the extracellular space to find a cell, if there's an end feed there that can take up glucose. And based on the paper of Bruno Weber, 99.8% of astrocytes in the gray matter, they have at least one end feed connected to the blood vessel. The second observation I call numbers. And this is a typical FDG the pattern of FDG signal in the human brain. And as you can see here, we have increased signal in cortical regions, in the brain cortex. And in the cortex, if we evaluate at the cellular level, we have 16 billion neurons and 60 billion non-neuronal cells, which means that we have much more glial cells in the cortex than neurons, which is a region that we have more signal, more glucose uptake. And in the cerebellum, where we have low FDG PET signal, we have the opposite. Much more neurons than astrocytes. And the third observation is related to the astrocyte neuron lactate shuttle, which was proposed in 1994 by Pierre Magistretti and Luc Pellerin. And it's a cooperation between neurons and astrocytes, where they propose that most of glucose is taken up by astrocytes, is metabolized to lactate and shuttle, and this lactate is shuttled to neurons. So neurons would prefer to use lactate as an energetic source. So based on these three observations, it seems that there's an important component on FDG PET signal associated to astrocytes. And we decided to test this hypothesis, and the first thing that we did, we decided to activate astrocytes. Decided to activate the astrocyte neuron lactate shuttle and see what happens in the FDG PET signal. And this is the average of 10 animals before activating astrocytes, and this is what happens after you activate astrocytes. And as you can see here, we have increased signal in the cortical region of these animals. We have this FDG hypermetabolism. Okay, but what we see in neurodegenerative disorders usually is hypometabolism, right? Would we be capable to mimic the FDG hypometabolism that we see in neurodegenerative disorders by only deactivating astrocytes? We did the same. We deactivate the astrocyte neuron lactate shuttle, and this is what happens. This is before deactivating and after deactivating astrocytes. So we consistently reduce the FDG PET signal in cortical regions. And we did that with a pharmacological GLT-1 downregulation in astrocytes. Then we did the same with the Krelox model. As you can see here, this is after selectively removing GLT-1 in astrocytes, and after the Whiskers stimulation, we can see this reduction in the signal in the barrel cortex. And we also did this with adenovirus injection, selectively removing the MCT4, which is a lactate transporter that shuttles lactate from astrocytes to neurons, and we see the reduced response in whiskers stimulation when we do this selective removal of MCT4 in astrocytes. So it seems that if we manipulate astrocytes, we change FDG PET signal. This is a very nice comment about our work from John Stonson, and as you can see here, he proposes that FDG PET signal is a composite of glucose that is inside neurons and glucose that is inside astrocytes. But why is this important? So what do we see here? What do we see in this typical hypometabolism that we see in Alzheimer's disease? Is neuronal dysfunction or is astrocytic dysfunction? So we really still don't know the answer. It's very nice to see that in this consensus paper in Lancet Neurology, they already acknowledge the role of astrocytes in the FDG PET signal. So FDG PET measures the intensity of brain neuronal and astrocyte reactivity. If you want to read more about the role of microglial cells in the context of FDG PET, this is a very nice discussion by our group and Matthias Brendel group in Germany, in which we wrote some comments and response to comments. A very nice discussion if you want to understand more about the role of microglial cells in FDG PET signal. And finally, we have been seeing a lot of data showing that astrocytes and microglial cells, they communicate. And this is a very important evidence showed by Shane Lillow and Ben Barris, in which they propose that neurotoxic reactive astrocytes can be induced by activated microglia. But this is evidence from animal models and post-mortem tissue. We decided to test this crosstalk in vivo by using biomarkers, and we use GFAP as a biomarker of reactive astrocytes and TSPO as a marker of reactive microglia. And as you can see here, if we divide individuals based on TSPO positivity, GFAP, it's only associated with A beta in TSPO positive individuals. And it seems that the TSPO effects of A beta and GFAP are closely related to the TSPO expression in the brain. And if we do an structural equation modeling to try to understand whether this association can explain a cognitive decline, we can see in TSPO positive individuals explain 76% of the cognitive variance. So this is something very nice. We have a student that is an artist. Now he does illustrations about our findings. So every paper that we publish, he does an illustration. And this is an illustration for this last results here. It's Marcelo Madrid, an MD student. And as you can see here, we have above one happy astrocyte, one happy microglial cell, but the neuron is already seeing what is happening in other brain region. Here we have a microglial cell telling to the astrocyte, okay, we need to fool the neurodegenerative cascade. So this is a very nice representation of the findings. So, as concluding remarks, based on the current evidence that we have, neuroinflammation in living individuals in AD is in fact glial reactivity. Glial biomarkers affect the ATN system. They affect all these ATNN labels. And interaction between glial cells may drive disease progression. We should think about glial cells as we think about neurons. So we have glutamatergic neurons, GABAergic neurons, serotoninergic neurons, dopaminergic neurons, and we have an homeostatic and an unreactive astrocyte or microglia. We need to think about them as multiple cells that present multiple different phenotypes that are coexisting all the time in the brain. Okay, there's one more thing, And we just published the first paper with ultra-sensitive blood biomarkers in Brazil. This is a paper validated with CSF biomarkers. It was out last week. And we are going to, as you can see here, they work very well. This is the first evidence in the Global South. So they are very good to differentiate cognitively impaired from dementia, cognitively impaired from AD, and to discriminate low and high CSF levels of A-beta 42 and 40. So they work very well as they work in the populations of the Global North. And we are running what we call the Brazilian Initiative of Blood Biomarkers, and we are supposed to have, in 18 months from now, almost 7,000 individuals measured with ultrasensitive blood biomarkers in the region. And of course, we are willing to collaborate. So my lab is based in Brazil, in the south of Brazil, in Porto Alegre, and everything that I show here was made by this team of students. The lab has now 42 students. It's a big lab, and they did all the work. I'd like to thank my collaborators and funding, and I'm happy to take questions.
Thank you very much for these great talks. I think we have now 15 minutes for a Q&A session. So is there any question from the audience?
Hi. I think that there is evidence robust enough for pushing forward anti-inflammatory therapies for AD. Might be the same case for PD. I think that there are two things that need to be properly balanced together. So on one hand, I guess it's critical time for intervention. So if you intervene very early with, let's say, you want to upregulate prime 2, you better go very early at the time of the amyloid deposition. If you intervene too late at the time of tau, this might be even deleterious. And another issue is the delivery route. So brain delivery is like real estate. So the only thing that counts are location, location, and location. So I think that a wrong combination of timing of intervention together with not an appropriate delivery route might well explain failures in clinical trials of anti-inflammatory compounds. That's my point, but I would like to know your thoughts about it.
Yeah, I can answer that. I completely agree with you. But I think that the problem is that the evidence that we have currently is based on glial biomarkers, right? If we check the evidence about like cytokines, interleukins, you can see that the evidence sometimes goes up, sometimes goes down, sometimes not changed. So I think that we really don't understand whether what we have is something similar to classic inflammation that we have in the periphery, right? The evidence that we have is all based on glial biomarkers. This is what I think that we measure with all these biomarkers that we are testing in vivo right now. But I agree with you. It depends on the disease stage. It depends on the brain region. So there's a lot to understand. But for the case of astrocytes, Marta showed before, if we check these new single nuclei papers, we have at least 8, 10 different astrocyte subtypes in Alzheimer's disease that they change based on the region. So there's a lot that we need to learn in terms of astrocytes and glial cells. There are multiple phenotypes, and we are still in the beginning to understand the functions that they have.
Well, thank you. Thank you very much. It was a fantastic panel. And as Eduardo, as you mentioned, GFAP is part of the new criteria, but there is no assigned a role in the ATN. If you were to assign a role, where would you locate GFAP?
This is a great question. There's a lot of people that say that it's a surrogate marker for amyloid pathology, right? But you can have increases in GFAP in the absence of A-beta pathology. So we have a paper that's not published yet that we can see like high levels of GFAP in VCI in the absence of amyloid pathology. So I think it's a marker of glial reactivity, but the way that I believe that we should use them as a marker of GFAP positivity. Because this is what we do for the other biomarkers, right? We measure amyloid with amyloid biomarkers. We measure tau with tau biomarkers. Then we measure astrocyte reactivity with GFAP. So GFAP is a marker of GFAP positivity. And I think that this happened before in the oncology field. We are going to move to characterize these biomarkers based on their names. So GFAP positivity, TSPO positivity, I think this is the best way to go.
So you think it can play a role in the staging of the disease to be more precise? And
Yeah. If we use, I saw a poster from your lab as well that you have this increased chance of cognitive decline in the group that is positive to amyloid and to GFAP at the same time, right? This is pretty clear for us right now. An individual that is positive to amyloid and positive for GFAP progresses faster than an individual that is positive to amyloid and negative for GFAP.
But,
Go ahead.
But and I mean, just to mention how complex is this, and in our paper where we validated with neuropathology, and this is in line with other papers of Gemma Salvador, the main correlation of GFAP is with tau more than A-beta. But of course, this is in a terminal stage of the disease,
Yeah.
so it's something that definitely is moving at, depending on the stage of the disease, you should interpret the biomarker in a different context, right?
So let me try to contribute with that as well. So Alexei Verkratsik, he always showed that an astrocyte can be GFAP positive in the morning and GFAP negative at night. So it's very complicated to do this like this neuropathological validation with astrocytes because they change phenotype all the time. They're very plastic.
I think there is another question.
So me? Okay. Well, thank you for
We have at least 3 questions, I think. 1, 2, and...
Okay, so thank you for all the talks. This is a question for Amaia. I would like to know for how long do they live, these human astrocytes, once they are in the mouse brain?
Sorry, how long?
How long do they live, the human astrocytes?
How long they last in the brain?
Yeah.
I mean, we have analyzed animals up to a year, and the astrocytes are still there. They integrate well, and they last for a long time. Yeah. They are there until 11, 12 months at least. We have these human astrocytes in the mouse brain.
Okay, thank you.
There are another 2, yes.
Okay. Yeah. Thank you very much for your really exciting talks. I was wondering, and I think this is a question that maybe each of you could give me a short answer because it's something that I've been thinking a lot about, and many people wonder about this. So could pathology-specific astrocyte states be driven more by their interaction with other cell types, like neurons, oligodendrocytes, microglia, and also could that depend on the pattern of neuronal activity that changes their metabolic rate and how they interact and function?
So sorry, I don't know if I get your question, but probably you ask about the interaction between different brain cells in the context of Alzheimer's disease and how they can affect each other.
No, I'm wondering whether, because in your talk, so what I understand is that there are different states of glial cells, like microglia and astrocytes, depending on the pathology and the progression of pathology, right? So now I'm wondering whether this is also driven, so these different states are also driven by how these glial cells are communicating, like intercellular communication with other cell types as well. And then the second question is more related to their metabolic profile, depending on these interactions.
Well, I definitely think that yes, this glial response to pathology is definitely affecting how the cells interact and how they contribute to the progression of disease. And it's probably not only depending on the states of the disease, but also the region of the brain, and if there are other co-pathologies. So there is really an effect of this glial response, let's see, to how disease progresses. And about the metabolic part, I don't know if you want to comment.
The short answer, they are, I believe, based on the data, that they are communicating all the time. You know, when a reactive microglia, when a microglia is reactive, probably the astrocyte will become reactive as well. We are doing a lot of selective removal of enzymes in astrocytes right now, and by removing like specific enzymes, we completely change the metabolism, the morphology of the astrocytes. It's very interesting. They become atrophic very fast.
Okay, thank you.
We have another question there, Mark. Yes.
Thank you. It was a great session. So it's a question for all of you about the future of glial biomarkers. So what will be the future? We will be using a combination of different biomarkers that define different states of microglia and astroglia. If so, how we discover them? Because so far what we have done is a candidate approach. We know TREM2, GFAP, OKL40 are of interest, so then we measure them. But can we discover new biomarkers in an untargeted approach that define different states? And really, how we validate them? Because there are dynamic changes, so is pathology the best way to validate them? What's your thoughts about it?
So I can start with that. So I think that we're starting to understand these subtypes right now. There are two important papers. One is, it's a paper by Green from 2004, a Nature paper, showing 9 to 10 different phenotypes. So they are there, like there's more than one reactive phenotype, I think 2 or 3 reactive phenotypes. The paper by Alberto Serrano Pozzo as well in Nature Neuroscience, they have, I think, 8 different phenotypes. 3 of them are reactive astrocytes. One of the reactive astrocytes that they described has none of the canonical markers of astrocyte reactivity. So I think now we're starting to understand these subtypes. Then we need to develop biomarkers that are indicating different astrocyte subtypes, as we did for neurons in the past, right?
So you will take the proteins defined by RNA-seq studies, and then as a candidate approach, just measure them, and then how you validate them, how you compare with?
Always very complicated. As we said, they are dynamic. Maybe we can find one that is not as dynamic as GFAP, and you have a very good association in CSF, blood, or PET with brain tissue. But it's always a challenge to do the neuropathological validation with these new glial biomarkers. But I will check these new subtypes and see if we can find a new marker that can be helpful in terms of adding information to prognosis or something like that.
And regarding soluble TREM2, Estrella, do you think, is it simple biomarker, or does it have a function, a biological function?
Yes, I think that is an important point, because I do think it has a biological function itself, because it has immunoglobulin domain, so it might have a biological function. So maybe one point, as I mentioned before, that why the therapies are not working is because it is lowering the soluble TREM2. And then we have to investigate the different forms of soluble TREM2 and know what is the biological meaning of these functions.
So maybe targeting soluble TREM2 is not a good idea for TREM2 agonists.
So maybe you have to supplement with soluble TREM2, or you have to modulate the expression of, not the expression, but the generation of soluble TREM2. There are some polymorphins that are modulating the generation of CSF soluble TREM2, and that they are protective, or maybe you have to go through other approach that is not stabilizing soluble TREM2, or is not affecting that much the possible function of soluble TREM2. So Yeah, and I do think that maybe the chronic stimulation of the TREM2 activation in microglia is not the appropriate way to go, but stimulating the TREM2 functions, but by other signal that represent more the protective functions that we have seen in the clinical cohorts. Right.
So I don't know if there is another question.
I would like to ask a question. So it's the function of TREM2 in rodents and humans the same? They do the same thing for in rodents and humans? Because we are discussing that, microglial cells, they are really different in different species, right? So
for example, one difference is the soluble TREM2 isoforms that I think, I don't know if any group is researching or focused on that, but I think that's very important and it's not trivial, and that is a main difference between rodents and humans, the soluble TREM2 isoforms. So I do think they may have a regulatory effect on the activation by TREM2 of microglial cells.
Mm-hmm. So thank you very much. I think we have to end now the session. Thank you for this very stimulating discussion. And we have now a coffee break.